Good morning, everyone. Welcome back to another day of the Longevity Leaders Conference. We've got a real treat this morning. We're going to be looking at the UK Spine Network in some depth and especially what it can bring to the longevity community. Just a couple of housekeeping points from me before I hand over to Chris, our moderator for this morning's session. Please do put your cameras on. We'd love to see your faces. This is supposed to be an interactive session, and it's great if we can see the people we're speaking with. So please don't be shy. Feel free to do that. Hi, Bev. Nice to see you. Please use the chat if you've got questions or comments to put to the group as the conversation is happening. Also, if you'd like to ask your question out loud, feel free to use the hand raise button, and then our panel can also call on you to speak out loud. But without further ado, I'm going to hand over to Chris, our moderator for today. Thanks very much, Chris.
Thank you very much, Angela, and good morning, everybody. Thank you for taking your time with us this morning. As we grow 1 hour older during this session, we'll be hopefully growing 1 hour wiser as well, listening to my colleagues around the table, Professors Liz Sapey, Bountra, and Sir Mike Ferguson, who will be talking about the concept of the UK spine, and I'll introduce that as we go through this starting prologue, and then talk about the individual vertebrae in the spine and the connective tissue that holds it together, and what it is that it delivers then, not just to the UK community, but to the worldwide community of medicines discoverers who are interested in the area that brings you to the table this morning too. So you are not alone. You have a vast array of people who are interested in this area, who believe in this area, who have the tools, the technologies, the insight, and the intellect to help you help those of us who are all aging 1 hour in the next hour. My name's Chris Molloy, and I am Chief Executive of the UK Medicines Discovery Catapult. What is the Medicines Discovery Catapult? It's the National Research Technology Organisation for Medicines Discovery, tasked with industrialising and driving the adoption of new tools and techniques for the sector. We are helping the sector prove new technologies. We're helping the sector drive new ways of working. And our 140 plus technologists and experts are spending time across the UK helping organisations help the industry. Now, one of the really important parts of this activity is translating academic endeavour with industrial rigour into products and services that the industry can consume, financiers can finance, and ultimately patients can benefit from. And there is no greater example of that than the work being done with my colleagues here within the UK Spine initiative, something that has grown over the last 4 to 5 years and is delivering now a porous pipeline of assets that are moving from concept through to the clinic. So before I introduce these esteemed colleagues of mine, the UK Spine consists of vertebrae that are academic centres of excellence, and the connective tissue that holds them together, the Medicines Discovery Catapult, is there to help harness with industrial rigour all of that academic endeavour and infrastructure and intellect in a way that bears the weight of a pipeline of new assets coming forward in this area of longevity. And why is this area important to do in this academic bounded way? It's important because industry finds this area a challenge. It finds this area high risk, but it recognizes that this is a future area of endeavor. And what it's looking for is organizations like ours and in the formation of the UK Spine to decrease the risk in this endeavor. New tools, new techniques, new trials. All of these combined reduce the risk of industry leaning in to this area of longevity, something which all of us hopefully will be beneficiaries of. Because whilst industry may say longevity and aging is not a disease, it is biology, and all diseases are biology. This may be a collection of or a combination of different types of biology seen in different organs at different times of our lives. But fundamentally, this is a biological problem that we are dealing with pharmaceutically. So without further ado, I will hand over firstly to Chaz, then to Sir Mike, then to Liz to talk through the UK spine, starting with early stage targets through drug discovery and then into trial, to introduce you to the vertebrae that take the load of this pipeline. And I will first move to Professor Chaz Bountra at the University of Oxford. Chaz, good morning.
Good morning, Chris, and good morning, colleagues. And thank you also to colleagues who have organised this event and put it on. So my name is Chaz Bountra. I mean, I've spent my life either in industry or academia worrying about coming up with new drug targets and then coming up with high quality clinical molecules and then taking those molecules into patient studies to demonstrate patient benefit. And whilst I was in GSK, I did that in sort of neurological diseases and inflammatory diseases and gastrointestinal diseases. And now in Oxford, we're doing it in a number of areas, in oncology, rare diseases, mental health, etc. But with this particular project, the spine project, what we recognize is that lots of elderly patients don't have one or two health conditions or morbidities. They have usually half a dozen diseases, and they are taking a drug for each of those diseases. And often they're taking 2 or 3 drugs to manage the side effects of those drugs. So they literally are taking a whole cocktail of drugs every day, maybe 7, 8, 9 drugs a day. And we now believe that biology is telling us that it may be possible to identify drug targets that will treat multiple morbidities. So we can have one drug to treat multiple morbidities. Of course, if we do that, then this will reduce polypharmacy, and I hope it may also reduce side effects experienced by these patients. Now, when we started this project almost 3 years ago, we hoped or anticipated that there might be lots of new drug targets out there for multimorbidity, and we soon realized that that was not the case. And so we have been building a very large national network with lots of academic centers, with patient groups, with SMEs, with charities, with big infrastructures in the UK, like the Medicines Discovery Catapult or the CRIC, and also with many, many colleagues inside the NHS. And we've been trying to bring all of this network of expertise and resources and talent and ideas together to sort of tackle this problem, because we believe it's a major problem for the UK, and it's a major problem for the world, and it's frankly a growing problem for the world. And so that's it. I mean, Chris, I think, outlined very nicely in this endeavor. What we've done is we brought together initially the University of Dundee, obviously with Sir Mike Ferguson, the Medicines Discovery Catapult in Manchester, colleagues in Birmingham, because they've got lots of clinical patient resources, a real focus on aging and translation, and a relatively new hospital with 1,300 beds and electronic records, et cetera, obviously colleagues here in Oxford, and then of course colleagues at the CRIC. But Beverly and her team over the past 3 years have really expanded that network to include lots of other universities and, like I said, patient groups and SMEs and so on and so forth, et cetera. So we now have a very broad network. And what we've been trying to do is to come up with this pipeline of new drug targets. And it was Mike Ferguson who actually came up with the idea of calling it a porous pipeline. And so the idea is that sort of colleagues can input ideas at any stage along that pipeline, and at any stage along the pipeline we can take outputs out and partner with pharma companies or VCs, et cetera, et cetera. So I think I've done enough talking, but sort of I'm very excited about this, and I'm very grateful to all of my colleagues who are on this call who have helped us progress as much as we have in the past 3 years. So thanks, Chris.
Thanks very much indeed, Chaz. And from targets, then come chemical tools, and ultimately leads, which can be taken on through and optimized. So we move quite naturally to Sir Mike Ferguson at the University of Dundee and the input that you and your group have, Mike, on this porous pipeline.
Thanks, Chris. Lovely to be here. So I'm Mike Ferguson. I'm Regius Professor of Life Sciences at the University of Dundee, and I'm a reductionist biochemist who has had to embrace the 21st century in terms of systems biology and systems medicine. It's always good to shift yourself out of your old comfort zones and into the latest challenges. And dealing with the multimorbidities of aging couldn't possibly be a better example of that. Within this consortium, the UKSPINE, what the University of Dundee really brings to the table is a translational engine in the form of our drug discovery unit, which is more than 15 years old now, and is about 120 people strong. And its focus is essentially to be a professional biopharmaceutical company, but embedded in a university and therefore not for profit. And it has all of the elements that you might imagine. It's got assay design and both biochemical and cell-based assay capabilities, mass spec-based assays, the whole gamut. But really importantly, and what's often missing in the rush to screening is follow-on chemistry, because if you don't have follow-on medicinal chemistry, really, you just move the barriers to translation one small step further. So we have about 45 medicinal chemists. Nearly all of these folk came out of pharma and biopharma, so it's a very professional entity. It's backed up, of course, by state-of-the-art computational chemistry and structural biology. We use a lot of structure-based drug discovery. And this is one of the reasons why we interface so incredibly well with our Oxford colleagues, with Chaz's colleagues, because they produce these things called target enabling packages, which is the crystal structures, the protein expression, 2 orthogonal assays, and starting point chemical matter docked to the crystal structures. And that's a fantastic start point for the drug discoverers in Dundee to do the hit expansion and lead optimization and so on. So we have all of those capabilities, and crucially, we also have in-house DMPK, so pharmacokinetic and efficacy models, so that we can iterate the whole process and focus in on quality medicinal molecules with appropriate solubility and so on very, very early in the process of selecting chemical series. So if I may say so, it's not a sort of amateur drug discovery entity, but it's a very professional one. It just happens to be sitting in a university. So think of it as a translational engine. We start where Chaz and et al. Stop, which is they develop the target-enabled packages. Where do those target-enabled packages come from? Just to go back one step, that comes from an incredible brains trust, actually, which Chaz has been very responsible for assembling in lots of relevant therapeutic areas to the multimorbidities of aging. But it now also involves open targets at Hinxton, and it involves the Medicines Discovery Catapult, John Overington and colleagues in Manchester. So we've got a tremendous machine learning, AI, and knowledge base coming up with innovative targets in the fundamental biological pathways associated, more or less. I mean, what I'm about to say is an incredible trivialization, but DNA damage and metabolic dysfunction, senescence, chronic inflammation, multimorbidities of aging. We need to be working in those fundamental pathways so we can find nodes where if we can modulate them or 1 or 2 of them simultaneously, we can affect not one but multiple morbidities downstream. So that's the concept. So I always joke to Chaz that the structural genomics end of it in Oxford and the drug discovery unit end of it in Dundee are the oily rag. We're the translational machine. The clever bit is in the selection of the targets. You then translate through to clinical quality molecules, and you get those into innovative clinical trial designs, which Liz is going to talk to us about. Why is it a porous pipeline? Well, if a member of the consortium, let's say a pharma company, has an asset which they've already developed, they don't need the front end. They just need the innovative clinical trials end of it. And similarly, something might start off as a brilliant innovative target, and translate through to a preclinical molecule, let's say, and a pharma company might say, we want to license that now and take it out. Okay, they can take it out of the pipeline as long as we can see that it's going to go somewhere. So this is the porosity, I think, is incredibly important, and why we are accreting new partners all the time. I'd like to emphasize that the UKSPINE is not a closed shop at all. All of the partners are really operating at risk at their own expense at the moment, just trying to get the proof of concept and get various programs off the ground. But we do look to accrete more and more partners as we will need to, and also to look to try and establish a public-private partnership of scale, so that UK can be a place where we do this and we do it well, and the outputs are patented in the UK, and therefore the benefits accrue to the UK system, not only in terms of treating our aging populations better and more humanely, but also if we're going to create new industries and new products, let's have some UK economic benefit from that as well. And of course, that's what Treasury will be interested in terms of the first P, the public part of the public-private partnership model. So in essence, that's what we see the spine doing. And my little bit with my colleagues is the going from target through to preclinical candidate status, and then of course using CROs through the Medicines Discovery Catapult expertise to clinical candidates, and then off to Birmingham and Oxford and elsewhere to do the appropriate trials. I think I've said enough now, so I'll hand on to Liz. I'll hand back to Chris, sorry.
Thank you very much indeed, Mike. So you're hearing how the UKSPINE, like a virtual pharmaceutical company focused on diseases associated with aging, is developing new drug candidates. And Mike's, I'll come back to Mike later on with some statistics for what's already moving through that pipeline. But from research through to development and to the University of Birmingham and Professor Liz Sapey. Liz, good morning.
Good morning, and thank you for the opportunity to talk about the UKSPINE and Birmingham's contribution to that, and also thank you to colleagues for joining us on this call. So I like to think that Birmingham closes this circle, if you like, enabling those amazing translational insights to then be turned into innovative clinical trials. So I'm a professor working in the Institute of Inflammation and Aging within the University of Birmingham. The Institute is perfectly placed within the new Queen Elizabeth Hospital, within UHB Hospital Trust, which is a hospital of 22,000 staff members and seeing 1.2 million patients per year. We're based within a clinical research facility with 80 whole-time equivalent nursing staff, and our whole remit is to understand how those discovery insights can be taken further down into translational insights and then turned into proof of principle clinical studies. So Birmingham has an innovative trial package design that we like to implement. If you come from the north, you'll call these bucket trials, south, maybe basket trials. But what we look at is to see how we can deploy these targets in the right population, looking at the right endpoints, the right biological fluid, the right clinical endpoints for patients. And we can then expand the arms within the trials to bring in new targets if appropriate, and thinking about that porous pipeline, drop targets from that particular line of inquiry if it's not having the effect that we would expect with the patient group that we're studying. Birmingham's a great place to do these trials. We have a fantastically diverse population. We're 42% non-white. We represent the gamut of social deprivation and wealth. So we can provide insights which can really reflect and have global relevance in terms of trial design and output. And we're really good at delivering clinical studies. So last year, even in the height of the COVID pandemic, we still had 12,000 patient visits coming through our CRF, enabling these discovery insights to move down that pipeline. So I think Birmingham offers that translational ability with our labs, our clinical trial design. And I just wanted to add that as a clinician working within frontline NHS healthcare, we have real skin in the game here. I was on call last night, so if my voice gets a little croaky, that's why. And most of the patients I saw were aging and not aging healthily. They were suffering from multimorbidity, and most of them were on more than 12 or 15 medications, which were doing them untold harm, as well as hopefully some good. being able to unpick that would have a huge benefit to our society, both nationally and globally. So I think I've got real skin in the game here to try and improve outcomes for patients by designing better studies informed by better translational insights and better targets.
Liz, thank you so much for closing the circle there for us. And you exemplify the problem very well there at the end. This is not a theoretical problem. This isn't some sort of spurious unmet patient need. And this is not an organ by organ problem. This is a holistic human problem that the world is now facing, and it's facing it, one day more every day. We are running up against a wall here of an aging global population, and somebody really needs to do something. Now, those of us who are in the spine decided that we could be those somebodies and put the skin, some of the skin in that game. But we come back to a few challenges that industry, and many of us have lived and worked in industry for much of our careers. We know industry faces in this space. Is aging a disease? Is it something that's reimbursable? Is it something that is understandable biologically? And I turn to a question before we get into other areas from Ian Newington. And I think if I could start with Chaz on this particular question, Chaz, because it's a sort of early stage biology issue, is do we really have a good enough understanding of the biology of some of these multimorbid or linked disease areas, cardiovascular and depression, for example? Do we really know enough about these pathways to be executing on that? Or do we need further validation, as the industry would generally put it?
Well, I think this is why, Chris, as you said at the start, this endeavor, if you like, it's too risky. We don't have novel targets. We don't have, if you like, good biomarkers for multimorbidity. If we're going to do clinical trials in some of the patients Liz was seeing last night, how are we going to stratify those patients? What are the regulatory processes? We need innovation at all of those stages, and that's why it's risky. There are publications coming out, and Janet and Liz have been involved in some of those, I think also Sir Mike, in terms of how some of these morbidities are clustering. So our knowledge on that is improving. But I suspect, Ian, the best way of illustrating it is that we do know that there are some very effective drugs in the clinic that are having benefits in multiple diseases. And so, for example, the example I always think of is anti-TNFs. Of course, these are largely antibodies. They've been used very successfully to treat rheumatoid arthritis, and they've also been successfully to treat inflammatory bowel disease. But, the person who invented those drugs, Mark Feldman, is absolutely convinced that those drugs, if they could get into the CNS across the blood-brain barrier, then they would be useful in a number of neuronal conditions, neurodegenerative diseases, etc. Because he believes that sort of the role of TNF and the inflammation cascade applies to a number of health conditions, etc., etc. But unfortunately, the reason they've not been tested is, one, well, because they are antibodies and they don't get across the blood-brain barrier, and secondly, because they're expensive. And if there was a small molecule and it was cheap, I'm sure it would have been tested in a lot of other diseases. The second example, Chris, and I know you're possibly going to touch on repurposing later on, and that's something that Bev and the team have done a phenomenal job of in the past 2 or 3 years. And this was not something that we initially thought of 3 years ago. But, taking the bisphosphonates, for example, these have been around for decades. They've been used to treat osteoporosis in millions, tens of millions of patients all over the world. There is now very large studies, retrospective studies involving tens of thousands of patients, that these bisphosphonates obviously reduce frailty and fracture risk, but they're also having beneficial effects in cardiovascular conditions. They're having beneficial effects in stroke. they're having beneficial effects in some inflammatory conditions. And there's also some recent data that actually, Chris, your team has been involved in with John Overington, that they may actually have utility as in some infections, antiviral infections, etc. So, there's an example of a class of drugs which have been in tens, hundreds of millions of patients all over the world in the past 4 or 5 decades. They are generic, they are very cheap, and they have clinical utility benefit in a number of conditions, etc. So I hope that answers your question, Ian, in a little roundabout way. So thank you, Chris.
Thank you, Chaz. And understanding the biology is a vitally important thing, because that underpins our likelihood of success. But being able to use that open data, the data in the real world, to see what does work and try and overlay that on the biology as a template, I think is a vital part of doing that. So you start either with the biology or you start with the real world. You end up in the place that says, right, this is what works. One of the areas that the aging and longevity space has suffered from industrially is people say, well, what are the models for this? How do I optimize anything against this set of conditions, or the aging of organs, or so on? It's all very complicated and difficult, and we haven't got a reliable model. If I could start with Mike, and Chaz, you may wish to come back on this, but Mike, in the optimization phase of what the DDU is doing for spine, how do you see the importance of models, either ditching the old ones or adopting the new ones, to that process of optimization?
Yeah, thanks, Chris. And I think Liz will probably want to come in as well. I mean, yeah, so models for aging. I mean, there's been fantastic work done using Drosophila, using C. elegans, and even yeast to understand the fundamental wiring diagrams of what associates with cellular and tissue aging. So it's not like we don't have, and mice, of course, as well. It's not like we don't have any models. I mean, Linda Partridge and colleagues have done fantastic work actually developing them. But always the chestnut is, how does that translate into the human situation, as we know? And Chaz, I'm sure, could give us chapter on verse from his GSK days, especially on the non-translation of animal models into human. So mice exaggerate, is the general modus operandi with animal models. And I think this is what's so important about spine. We have to take some gambles. So we have to use, based on knowledge and machine learning, we have to select really credible targets and develop the chemical matter which is sufficiently developed for to be safe enough, or to be safe to put into early human experimental clinical trials. And we're going to be, in other words, jumping into many extents from target into the human situation. And so I can't emphasize how important the experimental clinical trials are going to be in order to enable us to do that, because otherwise we could get sucked into that never-ending cycle of escalation of invertebrate and then animal models, only to find that it all falls over at the translational end. I'll hand over to Chaz first of all, and then Liz to comment some more.
Yeah, I mean, Mike talked about my time in GSK, and I can tell you that sort of, there were many times we had generated some beautiful molecules, and they worked beautifully in sort of disease models in animals. And then we took them into the clinic and they did nothing. And one of them actually we were developing as an analgesic, and it worked beautifully in animals. When we took it into the clinic, there was some evidence that it might have actually made the pain worse. So I'm afraid I'm not a great fan of animal models. And I much prefer, when we try and come up with new targets, to be biased by human genetic data if possible. Data generated in patient assays. We can access immune cells or cancer cells from patients and study those. I take a lot of confidence in that sort of data. And also, as Chris, you said, real world data. The bisphosphonate example I gave, there's data that it works in fractures, obviously osteoporosis, but it also has benefit in stroke, in cardiovascular conditions, in inflammation, cancer risk, et cetera, et cetera. So I think trying to pull all of that data together. Now, I appreciate, we need animal models to give us the confidence to carry on, but I put a lot more weight on some of those data that I've just mentioned.
Thank you very much, Chaz. And the acid test is in the patient, our ability to be predictive in the patient. And that's the key thing in drug discovery. And Liz, over to you for your views on this.
Yes, thank you. I mean, I think the wonderful thing about SPINE with the broad expertise we have across the program is that we can bring all of these elements together, right? So we can use the big data to give us an epidemiological signal of potential benefit with some of the targets, but we can use it in a smarter way through AI and machine learning to really identify the populations who gain the most, and just as important, the populations that either gain nothing or could be harmed from a therapeutic approach. We then have the opportunity to sample these patients and to look to see, is the biological signal of interest actually there? That to me is so exciting. When a therapeutic target of interest, you know that it's going to be focusing on a group of patients where that signal is elevated and ripe for targeting. And then we enrich that population in the proof of principle early phase clinical studies so that you know that you are likely to get a readout. And that smart selection of the target and the patient group who are most likely to benefit, and an awareness of those who aren't going to benefit from it, I think is the beauty of what SPINE offers here.
Thank you, Liz. And the SPINE seeks to de-risk for industry. It doesn't seek to replace industry in this. It's asking industry to lean in and say, what is it that the SPINE can do? What load can it take to de-risk this thing for you and produce molecules which have early stage efficacy in patient populations that matter, that are biologically understandable, and have enough proof behind them to be able to either invest more or collaborate more in the development thereof. Liz, but one of the areas that industry has found challenging in this space is, well, diseases associated with aging, aren't the trials going to take 20 years to do and have really sort of waffly endpoints to them that we can't really understand whether it's worked or not? That's not how this industry works. It's clean answers, clean regulation, clean use cases, and how do we, and the work of you and your group, help de-risk that for industry?
Absolutely. Well, you're right. There has been a tendency in the past for these studies to feel undoable because they would go on for too long. And some of the outputs that have been chosen for these studies have actually had little relevance to the biological pathway or process that we're studying. And that decoupling of the science from the patient population, I think, is where previous efforts have come undone. But this is what SPINE can address. So through the smart selection of the output that you're looking for, the signal that you're looking for, you can have rapidly either confidence in a target because the signal that you're expecting to see is there, the biological signal, or just as importantly, it's not there, giving you a clear signal that you need to stop, rethink, and try again. And the kind of studies that we've been delivering, looking at those biological endpoints that you know are associated with the disease pathway, you get readouts in very quickly, so you can agilely make decisions of go or no go based on those, and then develop that pathway further through your enhanced understanding of the disease. So by smart selections of targets and outputs, you can overcome some of these barriers really quickly and cleverly, I think.
Thank you, Liz. And where's the state of the art in terms of biomarkers in this space? You're talking about the biological targets themselves, but being able to measure these endpoints or these or the biological impact of your intervention. Where's the state of the art there, either in biomarkers or diagnostics for that matter? Have you got a view on that?
Yes, so there's been fantastic work that's done in this area, and I know that other members of the panel will be able to speak to this eloquently as well. But if you look at sort of immune metabolomics, cell function and phenotype, those sorts of things are giving very strong indications, clearly linked to mechanisms of aging and multimorbidity that can be used as targets. This is also informing our diagnostics as well, and with point of care and bedside rapid sequencing, we can look at those targets very quickly. So this is a fast-paced area of change, but one that UKSPINE is right on the crest of, if you like.
Thank you, Liz. I mean, all of this sounds like we are making diseases associated with aging an industrial reality here. Mike?
Yeah, just to follow up on what Liz says, the biomarker issue is absolutely crucial, not only so that we know whether any of our therapeutics are doing any good, but actually we're going to have to stratify people very early on. We need to identify people who are going to have a bad aging trajectory before they enter that trajectory. Trying to fix somebody of multimorbidities once they've got them all will be very challenging. But if somebody is in their late 40s, early 50s, who has, through a set of epigenetic, immunometabolic, senescence biomarker panel, a profile which says, this doesn't look good. Well, what's an analogy for this? High cholesterol and statins. So we have a solid biomarker for cardiovascular events, and we have very, very safe compounds which you can afford to give to people as preventatives. This is where we need to get to. It's not about making people live longer. It's not about letting them off the hook for their lifestyle choices, but it is about ameliorating bad trajectories by identifying the people who are at risk and tweaking their biology into a better place. If they also change their lifestyle choices, that's fantastic. But just like you wouldn't not give statins to somebody just because they don't have a training regime, you wouldn't not give somebody a helping hand if they're clearly, from a biomarker perspective, going to have a bad aging trajectory. That's where we need to get to. By 2050, 25% of the UK, and this is the same for most of the developed world, are going to be over 65. 1/3, 33% of our entire healthcare budget is going to be spent on those people. Now, we can't afford not to get better at it, right? We can't afford not to. None of the world can. Even, the world demographic is changing massively. You take a country like Brazil, it used to have an incredible pyramidal population structure. No longer. It's got a narrow bottom now, and a huge bolus of people moving up into old age. The world cannot afford not to do something about this. It is very difficult for pharma. It is too early, too risky, and that's where academic consortia should step in. That's what we're here for, in my opinion, to deliver for our stakeholders, the taxpayer and the medical charities. Thanks.
I could just add to that, Chris.
Okay.
I mean, and Liz and Mike have both mentioned sort of AI and machine learning. But if I could just illustrate this, sort of, when I first joined what was Glaxo in 1988, we were doing clinical trials then, and I remember doing one in irritable bowel syndrome. And in that trial, we made one measurement in each patient every week. So one measurement per week. We're now in the era when we look at sort of fluid biomarkers and imaging biomarkers and wearable devices, we can generate now tens of thousands of data points on each patient continuously, 24/7, longitudinally, non-invasively, and relatively cheaply. So for example, the company Somologics and the Spine Projects working with them, they can now measure 7,000 different proteins in a tiny sample of blood. And, and of course, imaging, in San Diego, they can now do a whole body image in 20 minutes for $600. And then the wearable devices, they can measure so many different parameters, and you can do it continuously, day and night, non-invasively, very cheaply. And then if we bring together machine learning and AI to make sense of all that data, I'm absolutely convinced it's going to come up with new biomarker signatures for different morbidities and new ways of stratifying patients. And that will de-risk how we do clinical studies in this patient group.
Thank you, Chaz. So, it's looking deeply through the data. These are complex issues. They may require complex medicines, but they are complex biology at work, and we need to be able to see through the complexity to the commonality that we can then treat. But you talked about measurement, and I think it's important for us to say, what are we measuring about what's going through the spine right now? This is a strong concept. This is UK PLC picking up the ball, running with it. How far have we got, Mike?
Switch my microphone back on. Yeah, thanks. David Bowker very kindly updated the numbers for me this morning. So, we've triaged quite a large number of targets that we've looked at really in depth, and a lot of these have been suggested from machine learning approaches through open targets and the MDC. But we've looked very serious, and then what we do is we do a very deep dive into the underlying literature and really pummel the concept. And so we've looked at so far 95 different molecular targets. I'd like to particularly mention Claire West's input into that. She's done a fantastic job together with John Overington's group and the Open Targets folks. So what have we done from then on? Well, out of those, we currently have 3 molecular targets in a drug discovery pipeline. So these have got crystal structures from the Oxford group and starting chemical matter, and they've been through high throughput screening, and they're in the hit validation and hit expansion phase, and the quality of compound series coming out are looking pretty good actually. And then at the other end of the pipeline, we've got 2 targets undergoing preclinical work. These are molecules that have come out of work of the Oxford Structural Genomics group with other clinical colleagues in Oxford. And then in the sort of area of target discovery, if you like, we've got really 4 major projects going on. These include the bioinformatics work that I've just alluded to, Claire and the MDC's work, but also we're sponsoring some whole genome CRISPR screening and some chemogenomics screening as well. So it's a portfolio which is growing all of the time. I just emphasize that this is being done by cobbling together the capabilities and goodwill and wherewithal of the current partners, not with some massive grant powering it. I mean, we have generously some money that funds the UK Spine from Research England, and matched by a little bit from the Scottish Funding Council, but this is not mega bucks money that we're talking about. So we're doing all of this pretty much on vapors. Thanks.
Thank you, Mike. And what your numbers there show, and the breadth of the things that you've just talked about, demonstrate the importance of a singularity of purpose. Groups of people who share that purpose, who come together and say, we're going to get this done and we're going to make progress, and hopefully others will follow. And I encourage all of those on the line, those listening live and those watching the recording, to recognize the community of purpose here, and approach and come and put up your hand and say, how can we as the UK Spine help you? How can you get engaged? Chaz, how would you like folks to get engaged with the UK Spine?
Well, Chris, I mean, sort of, we are, and I hope people appreciate this, incredibly collaborative. I mean, the fact that we've got so many universities and patient groups and SMEs working together with some amazing infrastructures in the UK, with lots of incredible clinicians inside the NHS. We are trying to do something that currently pharma I think would like to do, but it's not high priority for them. It's still too risky, etc. So they're keeping a watching brief on us, etc. But I think sort of please contact Liz or Mike or myself, or indeed yourself, Chris, Beverly, of course, and lots of colleagues in Beverly's team. I mean, sort of we're open to sort of collaborations. I mean, I think in terms of, Mike talked about sort of the pipeline that we've created, and it's been hard work and it's taken a lot of effort. But in addition, I mean, what we've done, or Beverly's done, is build an immense network across the UK. And in fact, the network is now becoming international because there are universities in Israel that want to work with us, etc., and we are collaborating with them. So I think that's a major achievement. And it's worth also saying that when we started this project, Chris, as you know, because you were involved, you know, we said we wanted to work together to create new targets for multimorbidity, come up with new biomarkers, new ways of stratifying patients. Ultimately, we were relying on Mike to generate clinical assets. And ideally, we wanted to generate de-risk clinical assets. In other words, assets that Liz had shown to actually have some benefit in patients. And we also wanted to come up with regulatory processes. You know, how are we going to do this in terms of regulation, etc. And, you know, and Beverly and the team have set up lots of collaborations. I mean, I think it must be well in excess of 40 different projects that are ongoing across Oxford with lots of partners tackling each of those things, you know, targets and biomarkers and stratification, etc., etc. And then, of course, I mentioned the repurposing stuff. I mean, wouldn't it be amazing if we could position a cheap, generic bisphosphonate, possibly as a new formulation to treat multimorbidity? And again, you know, Beverly and the team are looking to see, and, you know, if we can partner with some formulation experts to do that. And it's worth also bearing in mind with sort of bisphosphonates, as many of you are aware, currently to treat osteoporosis, they literally just have one injection once a year or once every 18 months. And, you know, again, for an elderly population, not to have to take a drug every day is such an advantage. So it's very exciting, and I'm just so grateful to Beverly. And I should also say, I mean, I think some of you know, Beverly's actually leaving us to go on to pastures new, etc. But we're going to continue working with her in some form or another. So thank you, Chris.
Thank you very much, Chaz. And the contact email is up on the chat for those of you who can see that. One of the things that has come through as a question, and it leads, Liz, from the thing you said earlier on, which is right drug, right patient, right time. One of the things both in MHRA's mind, also the innovative clinical design mind, is the right dose. So, you know, understanding that in the aged and multimorbid, the metabolic characteristics of the 75-kilo young fit volunteer man don't apply anymore. You know, how does that feature in your work and the work of the UK Spine?
Well, I mean, that's a brilliant point and a really important point to consider. You can only gain so much by testing compounds in young, strapping, fit people if you're then going to apply them to an older and multimorbid population. Fortunately, the Birmingham Clinical Trials Unit and the CRF have got significant experience in this, and we do very close dosing studies in the population where the medicine is likely to be used. So we can look to see what the appropriate dose would use, carefully monitoring people with side effect profiles to make sure that we are well informed with this information. And the most important thing to bring to this is our patients are well up for this. They really want to do something to tackle multimorbidity, not just for themselves, but for the future generations, as they know this is an expanding problem. So often a question that's asked is, how on earth are you going to get people to come in and be part of these intensive studies where we look at our dosing schedules? Well, we have lots of volunteers across all of our diverse community, giving huge confidence in the dose selection or the spectrum of dose selections that we go for once we've gone through this really important and careful testing phase.
You know, Chris, I'm absolutely convinced Liz was looking at me when she said young, strapping and fit.
I was.
Thank you, Liz.
And weren't we all, Chaz? But and implicit in the right dose question, Liz, is when it gets through to regulators, and you know, Ian's raised MHRA innovation, and they are most certainly open for business for those of you who've dealt with them. This is the sort of the soft side of the regulator, you know, there to advise, there to help, there to guide, not to judge. And, you know, in your conversations around clinical trials designs, you know, how are MHRA and other regulators starting to engage with this?
Really openly, actually. I think some people think that this is a closed shop and that it will be an uphill battle. And I'd like to reassure that it's absolutely not. We have a really useful and ongoing dialogue with all of the regulatory bodies across the UK and wider through our international trial arms to make sure that they are fully aware of what we're doing. They already buy into what we're doing. They see the vital nature and importance of it. And so we have these discussions about the approach we're taking to trials, the agility and the flexibility that we need, and within the careful safety parameters, which we are known for, they are really, really constructive and helpful. And we have not found that a barrier. We found them incredibly helpful.
Could I just comment on that, Chris, if I may? I mean, sort of, this whole project is very much about partnership. And I do think that, you know, when we set this project up, I remember Mike and Chris and yourself and Janet and colleagues, we were talking about why can we do this project in the UK and why is it maybe more difficult to do it elsewhere, etc. And we said, you know, we've got some great universities, we've got an amazing NHS, we've got brilliant infrastructures like the CRIC and the Catapults and the Sanger and so on and so forth, etc. And we've got lots of great leaders and innovators and entrepreneurs, etc. Those are some of the things that we talked about. But it's worth bearing in mind, I mean, sort of just what we've learned in the past year around partnership. You know, I'm sure you've all have heard of the Oxford AstraZeneca vaccine, etc. I mean, that was such a brilliant partnership between the university, a pharma company, government was involved, regulators were involved, public funders were involved. I mean, just an amazing achievement. And then it's also worth noting the Pfizer vaccine was first approved in the UK. And I think our regulators are absolutely brilliant.
Very good. Very good indeed. Thank you so much, everybody. You know, those of you sitting on the equator have moved in space by 800 miles since we started this conversation, and we've all aged by about 3,000 seconds. But I hope that that time has been well used, that that distance and that space has been well used, that you feel as though you understand what this UK spine has bound itself together with purpose to do, to de-risk, to bring together, to collaborate, to advance, and to enable the industry and that all of us who have a personal as well as professional interest in diseases associated with aging wish to see. Please do engage, tell your friends, tell anybody that is interested in this area that UK Spine exists and why it exists. I deeply thank my colleagues Liz and Chaz and Mike for your time today, but also for the efforts of you and all of your teams over the last few years. I know my team at the Medicines Discovery Catapult is really enjoying this program and feels its purpose very deeply. Folks, you've been stars this morning. I hope, audience, that you have been energized at the beginning of your day and will take that energy through your day. Very grateful for your attendance and look forward to being in contact with you. Back to you, Angela.
Thank you, Chris. And thank you to our panelists. I don't know about everyone else, but I'm feeling really energized. That hour just flew. I don't know where it's gone. So thank you for such an interesting and insightful set of presentations. Look, thanks again for joining us all. We hope you'll stay with us for the rest of the day. If you're interested in more of the innovation and support that's coming out of the UK, we have a session up next from the National Innovation Centre for Ageing. So please do join us and get involved there. But I'd like to say once again, thank you to our group from UK Spine for today's session. Thank you all for watching, and we'll see you again soon.